Good morning. So today I will talk about the Streptococcus agalactiae that causing disease mainly in cattle and also other species. As you know that the Streptococcus tuberis was grouped as a group B Streptococci, and it caused disease as we call it invasive neonatal infection in newborn, in human, and also in adults, mainly with women. Approximately 30% of healthy women around the world, we can find the colonization of this bacteria. And of course, Streptococcus agalactiae has been recognized as a major contagious mastitis pathogen causing mastitis in dairy cows. And other animals, especially pet animals, like dogs and cats, and also cause outbreaks in different kinds of fish, in both freshwater fish and marine fish. In northern Thailand or in Chiang Mai specifically, we used to survey the bacteria causing bovine mastitis in Chiang Mai and other provinces in northern Thailand. And as you can see from the picture that almost half of the bacteria that we found, they are in the genus Streptococcus. And the most common one is Streptococcus agalactiae, followed by Streptococcus tuberis. In humans, there's a previous report in Chiang Mai about the prevalence of the patients admitted in the hospital in Chiang Mai that infected with Streptococcus agalactiae from 2006 to 2009. As you can see that there's an increasing prevalence of in each month, the increasing prevalence of infection was increased in this period. So it seems like Chiang Mai or in northern Thailand, we have the high infection of Streptococcus agalactiae in both human and in cattle. And also there are many reports of outbreaks of these bacteria in fish around the country in Thailand. So the objective of this study was to investigate genetic variations of Streptococcus agalactiae strains isolated from bovine mastitis cases and compare them with isolates from fish and humans. In this study, we recruited 27 isolates. They're all from Chiang Mai, only one province in northern Thailand. During 2011 to 2014, 17 isolates were from bovine mastitis cases, 5 from tilapia cultures from different farms, and 5 isolates from septicemic human patients in Chiang Mai province. They all previously confirmed to be group B Streptococci, and they all kept frozen at -20 in BHI with 5% glycerol until regrown for the further analysis. And then after we regrown in the media, we use the PCR to confirm all the isolates again, to confirm confirm them as Streptococcus agalactiae. And then we perform another multiplex PCRs to do the capsular gene typing. Then we use the pulsed field gel electrophoresis analysis to primarily genotype these isolates using the Smal 1 or SMA1 resistant enzyme. And then we compare the DNA fragments and interpret them using the criteria described by Tenover in 1995. And we also use a program or software to analyze the picture of the pulsotypes that we found among all 27 isolates. Then we also perform the multi-locus sequence typing, or MLST, by doing PCRs to amplify the 7 housekeeping genes and then send for sequencing. Then each isolate were assigned a sequence type based on the allelic profiles. And then we created a minimum spanning tree to illustrate the genetic relationships among these isolates using the MLST system. So this table shows the results of what we found among the 27 isolates. First, most of them are isolates that compared to the sequence types that are already there in the database, they're all there already, but we found, we reported one new sequence type, which is sequence type 652. This isolate come from human. Among the bovine isolates, all 17 isolates are the same, which is a sequence type 103 and capsular type 1A. For the fish isolates, all 5 were also the same, sequence type 283 and capsular type 3. However, for the 5 human isolates, there are more variations in these, among these isolates. We found the new sequence type, which is capsular type 2. We found one isolate that similar to the bovine isolate, which is sequence type 103 and capsular type 1A. We found 2 isolates that similar to the fish isolate, which is sequence type 283, capsular type 3, and another isolate that different from others, sequence type 1 and capsular type 5. When we use PFGE, or post-future electrophoresis, this figure shows the analysis of the patterns that we found from DNA fragments among the 27 isolates. The dashed line represents the 80% similarity index, which is correspond to closely genetic relationship based on turnover criteria, or only one band different. So using this cutoff or threshold, we can cluster some isolates into 4 different similarity clusters, A, B, C, and D. Isolate from cattle are members of cluster A and B, even though they are the same capsular type and the same sequence types as we talked before. And cluster C are mainly the cluster of the fish isolates, but there are one member from the human isolates, which is same sequence type and same capsular types. And the cluster D is another cluster consisting of the one isolate from bovine milk and another isolate from human. And this figure shows the 2 minimum spanning trees based on the sequence types that we found among the 27 isolates, which is only 4 sequence types. In figure A, we label, each color or each circles, the size of the circles represents the number of the isolates. And in figure A, the color was labeled for each capsular type. And for figure B, the color label for source of the isolate. As you can see that we didn't see any sequence type variations within each capsular type in this study. So only one sequence types are the member of each capsular type. For in figure B, as you can see that we found that some human isolates share the same sequence type with bovine and fish isolates. So from our study, we found that a strain of human Streptococcus agalactiae had an identical capsular type, which is type 3, and sequence types 283, and also closely related in according to the PFGE pattern to strep egg isolated from tilapia cultures or from fish. In this point, we question, can it some kind of relate for this bacteria to cause foodborne disease in human? And there's some reports about that. In 2009, the previous report, Streptococcus agalactiae septicemia caused by consumption of contaminated food items in France. And recently, in 2017, there was a report investigating the outbreak of Streptococcus agalactiae, sequence type 283, and confirmed it as a zoonotic pathogen related to the consumption of freshwater fish, which caused a huge outbreak in 2015 in Singapore. About the bovine isolates, we found that the most common sequence type is sequence type 103. And this sequence type was occasionally being isolated in several species, including humans, cat, guinea pig, and also dairy cattle. In our study, one human isolate of this sequence type, we found it's the same sequence type as the bovine isolate, but have the different PFGE pattern. However, another human isolate has a close genetic relationship with one bovine isolate using PFGE, but different sequence types. So there's some unambiguous to use either PFGE or MLST to differentiate between some isolates of sequence type 103. So we think that both techniques should be used for genotyping Streptococcus agalactiae to maximize the discriminatory power for genotyping these bacteria. In conclusion, in this study, we confirmed that strep egg isolated from animals were more conserved within species than humans. So in humans isolate, there are more variations. And diversity of genetic materials can be detected using MLST with the presence of new ST or ST652. And all in all, in northern Thailand, cross-species transmission can potentially occur, and genetic investigation of a pathogen using molecular tools will help to identify the mode of transmission and improve control and prevention of such infectious disease, especially for Streptococcus agalactiae, which is kind of neglected species in human infectious diseases. We would like to thank the funding agency from Chiang Mai University, and we would like to thank about the technical supply from the Medical Science Research Equipment Center, Faculty of Medicine, Chiang Mai University, and the Central Laboratory Faculty of Veterinary Medicine, Chiang Mai University, Thailand. And thank you very much for your attention.